And that's something to consider because we have so many people now, and as I said, as the land masses get smaller because of climate change and the ocean rising, this is going to be problematic. Still, they have what we call preferred portals of entry. And this is why most microbiology textbooks will divide up diseases into categories based on parts of the body. It's because the microbes actually infect specific regions of the body. They have to be able to attach. We talked about receptor sites for viruses. They have to be able to bind with a specific cell type or a specific tissue type. The same is true for bacteria. That's where they're going to get in. They can only bind certain types of tissues, and so they can only get past certain barriers. There are some bacteria to think about. Turns out they also have preferred portals of exit, but right now we're talking about them getting in and getting out. So typically, if you look at microbiology textbooks, as I said, they're divided into regions of the body. Most of these regions involve mucous membranes, so organisms can get in via the respiratory tract, or they can get in through the gastrointestinal tract, or through the urinary tract, etc., etc. There are some pathogens, however, that can penetrate dry skin, and we've had some experience with things that we've talked about mostly in the area of our multicellular parasites. Do you remember which ones of those can go through dry skin? Oh man, that's way back there.
That's the
Hookworms. So hookworms can, usually fairly tender dry skin, but hookworm larvae, they're in the soil, and they can go right through dry skin. The Cercariae of blood flukes can go through skin. Well, I say dry skin, but typically they're in water, so I suppose the skin is wet when they go through. Still, they're going through what would be a dry skin surface. What else can go through dry skin? Well, Staphylococcus can. And they live on the surface of the skin, and so they can, under certain circumstances, go through the skin. So schistosomes, the blood flukes, Necator, one of the types of hookworm, but Staphylococcus can, also Treponema pallidum, which is causative agent. And probably other spirochetes. There are other spirochetes associated with water, where when people fall into water, again, it's a wet situation, but they go through dry skin. There are some types of pathogens that require what I call the parenteral route. And as soon as I say that, I think, is it pronounced parenteral or is it parenteral? And I don't know. Anybody know? It's a clinical term, I think. But it's like, it doesn't have to do with your parents, so I'm not sure. It has to do with some kind of damage to the skin. So they can only get through skin if the skin has been injured. A bite, a cut, a puncture, surgical procedure. So I have some lovely pictures. There's somebody receiving a bite and somebody else. I'm not sure what they did to that foot, but it looks like it involved an axe. Not nice. Anyway, the other thing we know is that some types of pathogens are vector transmitted, so they get through that first line of defense, typically dry skin, with the assistance of some type of arthropod. And so we talked about those guys in laboratory early. We had ticks, we had fleas, we had lice, we had mites, mosquitoes, tsetse flies, etc., etc. So the organisms have to be able to get in. That's their first thing they have to do. The second thing they have to do is be able to either avoid or cause damage to the immune system, immune cells. So it turns out they have a variety of ways of avoiding immune cells, and some of them can prevent damage caused by these cells, or at the extreme end, they can actually damage the immune cells themselves. So we talked about fairly early on the glycocalyx. The glycocalyx is a layer outside the cell wall on certain types of gram-positive and gram-negative cells, but they produce mostly carbohydrate. That glycocalyx tends to prevent bacteria from being ingested by phagocytes. Apparently, phagocytes recognize capsule material as the same kind of polysaccharide that might be produced by the body, so they don't see that as something foreign, and they will tend to ignore those cells. That's why when we talked about Streptococcus pneumoniae causing pneumonia, it's the capsule-forming straps that cause pneumonia. If they don't produce capsules, they tend to be eaten up by phagocytes and taken out. There are some other types of surface proteins associated with some bacteria. So Staphylococcus aureus produces a type of protein called A protein. It does the same thing as a capsule. It makes these cells resistant to phagocytes. Streptococcus pyogenes, another important pathogen, also produces a surface protein. These are called M proteins. So A proteins and M proteins work like capsules to protect cells against phagocytes. Some types of bacteria can be consumed by phagocytes, and even after they're consumed, they don't die. So typically, phagocytic white blood cells would consume organisms and then digest them using their lysosomes. Certain organisms, such as Salmonella enterica, various serotypes. So this says serotype types, but it's not the only one. Several different strains of Salmonella enterica can survive inside of phagocytes, and Mycobacterium tuberculosis also survives inside phagocytes. Well, this was something that people were really concerned about, and still people are concerned about this because our phagocytic white blood cells behave a lot like amoebas in the natural environment. And so now what people are doing is they're studying amoebas and studying the types of bacteria that can survive being ingested by amoebas, and thinking, some of those organisms might also be more dangerous inside our bodies, because if they're consumed by phagocytes, they don't die. So they wanted to know why. Why does mycobacterium not die when it's ingested by phagocytes? And what they found out was that the mycobacteria somehow prevent the transport of hydrogen protons into the phagosomes. If you can't acidify the phagosomes, then the digestive enzymes don't work. It's kind of like digestion in the stomach. You have to have that acid environment in order for digestion to take place. So these guys can just live inside the phagocytes. And this is very problematic because the phagocytes travel and move around the body, and they take those bacteria with them. Sometimes they fuse, and they can carry them around. We also saw Listeria. Remember the cells that we're using to actin to propel themselves through cells? They can get inside of phagocytic cells, and they don't die. They move around inside of those things. Then there are bacteria that can cause damage to phagocytic white blood cells. There's a type of enzyme called leukocytin. Kind of sketchy on this one. Is it an enzyme or is it a toxin? Most of the time, if I'm mentioning enzymes, I've said enzyme names end in ASE, except for cyclone, that don't. But anyway, leukocytin is a toxin/enzyme produced by certain types of bacteria, and it kills white blood cells. So both Staphylococcus aureus and Streptococcus pyogenes can produce these leukocytins. So they not only can they avoid phagocytes with their A proteins and M proteins, they can also kill phagocytes. So they produce leukocytins that do this. Staphylococcus aureus produces a leukocyte called Fanconi-Valentine leukocytin. It is a protein exotoxin, and like many protein exotoxins, so these are things secreted by living cells, these are encoded by viral genes. And the process is lysogenic conversion. When bacteria start expressing viral genes, the viruses have gotten into the chromosome as a prophage, and then they stay there for an indefinite period of time. And then, of course, there's HIV, which just takes out the T4 lymphocytes. We talked about how they do this. When they're going out of the lymphocytes, they make holes in them. This is going to cause the T4 cells to die. Without T4 cells, your immune system fails, because you can't get clones of B cells to produce enough antibody to do you any good. You can't get your T cells, your other types of T cells, to reproduce in quantity, and so your immune system fails. You can notice that there's some overlap here as we talk about how these organisms can cause problems. So they have to get in, they have to avoid the immune system or possibly damage the immune system. The next thing they have to be able to do is reproduce faster than the body can get rid of them. This is why you can take antibiotics that don't kill bacteria. They simply prevent them from growing. You can use static antibiotics because if you can get the organisms to not grow faster than the body can eliminate them, then the body's going to win. You have phagocytic white blood cells that are trying to take these things out. You have other mechanisms to get rid of some pathogens, NK cells and so forth. And so if you can just keep the organisms, the pathogens, from reproducing long enough, then oftentimes you can avoid disease symptoms. So from a pathogen standpoint, you want to be able to grow faster than the body can get rid of you. Well, this depends on a lot of factors. We know that growth is influenced by temperature, gas requirements, pH requirements, nutrient availability, osmotic pressure, etc. inside the body you have a pretty high nutrient load, but certain types of organisms, for instance, Clostridium organisms, they're primarily anaerobic. So they tend not to grow in healthy tissue because healthy tissue has too much oxygen. It's not a suitable place for them. And certain types of organisms are really not invasive at all. They just colonize a surface and then produce toxins that go into the body, and those cause problems. So generally, if microbial reproduction is slow, immune mechanisms will tend to eliminate organisms. On the other hand, if they can hide inside phagocytes, which Mycobacterium can do, this is probably why Mycobacterium tends to cause chronic illness. They're very slow growing, but when they're consumed by phagocytes, they don't die. And then they just hang out in there and get carried around inside the body. They can actually reproduce inside phagocytes. The next thing the pathogens have to be able to do is cause some kind of damage. They have to be there, they have to avoid the immune system, they have to grow and reproduce faster than they're eliminated, and then they have to cause some kind of damage to the host. Here we see some gross pictures of pathogens causing damage. You can find lots of ways in which they cause damage inside the body. And this is an area that's much bigger than we can cover. Again, this is an introductory class, so we're only going to look at a couple of examples of things that these organisms can do. Generally, there are what we call virulence factors, and virulence factors are either enzymes or toxins, although, as I said, sometimes the distinction between them is a little hazy, but they are going to make these organisms virulent. And as I said, you can have the same species of bacteria, but if it's missing the virulence factors, it's not going to be a pathogen. We have isolated Bacillus anthracis in this laboratory, and there was no suggestion that the organisms we were working with were disease-causing. On the other hand, it's probably something that somewhere somebody in an authoritative position would object to the fact that we were working with Bacillus anthracis, even though they weren't pathogens. So let's look at some important enzymes. For this class, because we said enzyme names end in ASE, I'm only going to put into the category of enzymes the terms that actually end in ASE. So we have enzymes called kinase enzymes. Do you remember what kinase enzymes did relative to metabolism? They moved phosphate groups, right? Kinase, also called transferase enzymes, but kinase enzymes move phosphate groups from organic compounds from one to another. When you look at bacterial kinases, I'm sure it has to do with phosphate transport, but what they do is cause the breakdown of fibrin. Fibrin is a connective tissue material. If you remember my explanation of inflammation, around the area where the blood flow has brought in a bunch of phagocytic white blood cells and antibodies and so forth, typically the body tries to wall that area off by depositing fibrin. And oftentimes this is how the body tries to prevent pathogens from spreading into the circulation in general, is to kind of coat them with fibrin. If the bacteria can produce kinase enzymes, they can break the fibrin down, and this is going to increase their invasiveness. So it's good for the bacteria. Turns out, this is one of those things that people have recognized. Bacteria produce a toxin, or in this case an enzyme, kinase. Streptococcus pyogenes produces a kinase, which is called streptokinase. We know it breaks down fibrin, and so people have been able to actually use this in a clinical setting. It has to be administered within a specific period of time after an incident occurs, but it's used to treat both stroke patients and patients suffering from heart attack, because those are typically caused by blood clot, and the blood clot involves fibrin. If you can get that kinase in there quickly enough, it'll break down the fibrin and solve the problem. Another enzyme is called hyaluronidase. Now, hyaluronic acid is a substance that tends to hold cells together. You can think of it as being like a cellular cement. So if the bacteria produce hyaluronidase, they can break that down, and again, it's going to increase their invasiveness. They can go through cellular layers. Think about something like the endothelium, where you have very thin cells, but they're connected to one another. They make the walls of blood vessels. If the bacteria can break down hyaluronidase, they can go right through those walls. Another type of enzyme is collagenase. Collagen is one of the primary components of dense connective tissue. So if they can break down collagen, that means they can go through connective tissue barriers. And again, this is going to increase their invasiveness. So kinase, hyaluronidase, collagenase, all of these tend to increase the invasiveness of bacteria, allowing them to get to spread through the body and not be walled off. The body can't protect itself as well. Another enzyme that we worked with in the laboratory, or we tested for in the laboratory, is coagulase. Coagulase is an enzyme that's produced by Staphylococcus aureus. I doubt that Staph aureus is the only type of bacteria that produces this. It's just the only one we tested. Anyway, so they produce coagulase, and it coagulates blood plasma. So it causes clotting. Now, we just talked about breaking down fibrin to increase invasiveness. What is the staph doing? Apparently what they do is they kind of wall themselves off. This is going to prevent phagocytic white blood cells to getting to where they are, so they can't be consumed as readily. And they tend to form these masses of staphylococcus, and then there will be dead phagocytes and so forth in the area. A lot of pus associated with Staphylococcus aureus. Staph aureus is one of the organism types that is pyogenic. Pyo means pus forming. There's a couple of other enzymes that are used for diagnostic testing, DNase and lecithinase. So you can actually test whether bacteria can break down DNA or break down lecithin. Neither one of these are ones that we use in the laboratory. So never mind. Okay, so those were enzymes. And again, I'm sticking with enzyme names ending in ASE. If they have a different ending, then we're going to call them toxins. If the toxins are exotoxins, this means that they're proteins and they're being released by live cells. So they're being dumped into the environment, just like exoenzymes. Exotoxins are going out. Most of these are acquired through lysogenic conversion. So they're actually encoded by viral genes that the bacteria have picked up. The bacteria are carrying prophages. Turns out that the leukocidins that are produced by both Staphylococcus and Streptococcus are exotoxins. And so they are the result of lysogenic conversion. And again, leukocidins kill phagocytic white blood cells. They kill other types of leukocytes as well. Then there are toxins called hemolysins. Many bacteria produce more than one. Hemolysins kill red blood cells, erythrocytes. Staphylococcus aureus produces an alpha hemolysin and a beta hemolysin. Streptococcus pyogenes produces streptolysin S and streptolysin O. So they have more than one type that they produce. And again, this makes them pretty nasty pathogens. They can kill your white blood cells. They can also destroy your red blood cells. They get into the bloodstream, they can cause a lot of damage. Some of the toxins produced by Staphylococcus aureus are referred to as pyrogenic toxin superantigens, or PTS. These are responsible for causing both Staphylococcus food poisoning and toxic shock syndrome, which is usually caused by Staphylococcus aureus. Superantigens. Well, what the heck are superantigens? We learned that antigens are foreign agents. They can be molecules, they can be cells, they can be viruses. They get into the body and they trigger an immune response. Well, typically, an immune response is pretty small. You have lots of different cells, but only a small percentage of those cells are activated by any typical antigen that gets in. When superantigens get in, they trigger a huge immune response. What they do is trigger a whole bunch of helper T cells. They then start releasing their cytokines in overabundance, and this causes a lot of damage in the body. So you get a massive immune reaction. It's not specific to any antigen, and unfortunately, some immune cells can cause significant damage. We know that the cytotoxic T cells can do this, but if you activate a lot more phagocytes than you should, then you're going to get more of the substances phagocytes produce than you should, and some of those cause damage too. I thought these numbers were kind of interesting, that in a normal immune response, you're only releasing, or you're only activating about 0.1001 to 0.001% of the body's T cells. Very small percentage. Normally, and as I said, you have lots of subpopulations of T cells, but you're only maybe triggering the activity of one subpopulation. So it's a very small percentage of cells that are activated. These superantigens will trigger activation of much higher percentages of cells. So you have very large numbers of T cells active. They begin to produce and release cytokines, things like gamma interferon, which activates the macrophages. And then, as I said, the phagocytes can move around. They can produce more complement. They can consume cells. They can release some other substances besides complement. Complement, of course, makes holes in cells. And then the T cells can release, if they're killer T cells, they can release granzyme and perforin. If they're helper T cells, they can release some of the other substances. What was it? Tumor necrosis factor they can release. All these things are just abbreviated in here. Anyway, this can cause severe damage to tissue. Then we have some toxins that are much more specific. A lot of bacterial toxins are going to specifically target a particular cell type and have a very specific action on that cell type. The Clostridium toxins are like this. Clostridium toxins, again, are acquired through lysogenic conversion, so they are encoded by viral genes. We know that there are 2 different types of Clostridium that were in our outbreak list, which reminds me, for the Clostridium organisms that you need to know for the matching section that's on the final, we're going to add one more that we did not talk about in lecture, and that's Clostridium perfringens. Clostridium perfringens is the Clostridium that causes gas gangrene, and they can be extremely damaging. But Clostridium perfringens. Of course, we also didn't spend any time talking about Clostridium difficile, which is also nasty. But back to the toxins. So Clostridium toxins. Clostridium tetani causes tetanus or lockjaw. The term tetanus means sustained muscle contraction. If you do physiology experiments with the physiographs, if you get a muscle to go into tetanus, it will contract and maintain that contraction. Tetanus is sustained contraction. Meanwhile, Clostridium botulinum causes flaccid paralysis. So these toxins that are produced by these 2 types of Clostridium appear to have exactly opposite effects, and they appear to affect muscle. But it turns out neither one of these toxins act on muscle at all. These are neurotoxins. They have been analyzed, and it turns out that their amino acid sequences are very nearly identical, and sometimes they cross-react. So sometimes the botulism toxins in certain individuals will have an effect more like tetanus toxin, and vice versa. The tetanus toxin is sometimes called tetanospasmin. This is the toxin that causes tetanus or lockjaw. And the reason it was called lockjaw is because if a person is really suffering from tetanus, their skeletal muscles go into a state of sustained contraction. Their jaw muscles would sometimes contract so tightly that they would break their teeth. They cannot relax their jaws. People are in these really stiff positions. It would be like having a muscle cramp body-wide. They're all doing that. Excruciatingly painful, and they don't let go. Well, so what's happening here? It turns out that tetanospasmin isn't directly causing muscle contraction. Instead, what tetanospasmin is doing, it's acting on inhibitory neurons. Inhibitory neurons produce 2 types of neurotransmitter, gamma-aminobutyric acid or GABA, and glycine, which is an amino acid. These inhibitory neurotransmitters have to balance excitatory neurotransmitters. If you don't have that balance, you don't have controlled muscle activity. Instead, any excitatory signal that comes to the brain is just going to be excitatory. There's no inhibitory to stop it. And that's why when the tetanus toxin is in your system, you lose any inhibitory signals, and all you get is sustained contraction. Excitatory takes over. These are some images. The one is showing an adult individual in the throes of tetanus, where the muscles are contracting. The other one is a little baby having tetanus. Again, the muscles are contracting body-wide, the limbs are stiff, the back is stiff, the jaws are clenched. This is most often fatal. I was visiting a physician to get a tetanus booster on one occasion, and the doctor asked, had I ever had any experience or maybe I asked him, I don't remember which way it was, but anyway, this doctor had actually had a patient that had tetanus. And the treatment, what they had to do with this patient was induce a coma, put the person in a coma and maintain that for 6 months while they administered antitoxin. They actually pulled the patient through. It's usually deadly, which again is why you want to make sure you keep your tetanus toxoid up to date. You need to make sure you keep getting your tetanus boosters because the amount of tetanospasmin required to kill you is significantly less than the amount required to initiate an immune response.
So when you're pregnant, you get a tetanus?
Huh?
So when you're pregnant, you mean to get the tetanus shot?
Anytime, if you injure yourself, you know, like some people that work outdoors, they're working with metal, step on nails, that kind of thing. If you injure yourself and you haven't had a tetanus shot within 5 years, I think you have to get one.
But so what the pregnant woman gets, it, does it cross the placenta to, I guess, give it to the baby or no?
I don't know.
No.
Get the Tdap so that it protects them against what they call
Yeah, I think they have to get their own, but I'm not sure if it does anything for them. I've often wondered about that also. If you're outside a lot and exposed to the tetanus toxin, if you had a booster initially and then you get exposed to the toxin kind of randomly throughout your lifetime, would you tend to produce your own anti-tetanus antibodies? I don't know, but it's not something you want to mess around with. So we have a really good vaccine. You need to make sure that you keep it up to date. The botulism toxins, as I said, cause flaccid paralysis, but they don't act on muscles either. They act on excitatory neurons, and they block the release of the excitatory neurotransmitter acetylcholine. Acetylcholine has to be released in order for skeletal muscle to contract. So if you can't release that transmitter, your skeletal muscles do nothing. And that's what happens under the influence of botulism toxins. I think there's 3 different types of botulism toxins. They're some of the most toxic substances known to humankind. Consequently, wouldn't you know, some people inject themselves with it. Well, actually, they have somebody else inject themselves with it. What you see in the image on the left are the Clostridium botulinum cells. Notice they have endospores. They have swollen sporangia. They're terminal endospores with swollen sporangia. And there you see an infant suffering from botulism. Completely limp, flaccid paralysis. Here we see somebody getting a Botox injection. This was initially used to treat people for muscle tics, often in the face. Sometimes the facial muscles would just start to contract rather randomly. It's really irritating and can interfere with vision and that sort of thing. So people started using Botox to stop the contraction of those muscles. It was very specifically placed. And then people started doing it to eliminate smiles and because they didn't like the wrinkles that were being caused. But it's dangerous stuff, Botox. It's botulism toxin. Where you would normally find this and where it's really problematic is in association with canned foods. When people started putting food into containers, if they were canning foods that were not acidic, meats and vegetables, if they didn't have a high acid content, they had to make sure that they were properly pressure cooked, pressure treated. That's what pressure cookers were for. If you just boil them and seal them shut, the botulism spores can be alive in them. We know that endospores are resistant to heat. And I often wonder about this because now there's quite a number of like health food stores will carry various types of olive oil that have got vegetation stuck in there. And I wonder, or vinegar would be fine, but olive oil, that's not acidic. And could the vegetation have botulism spores associated with it? Why not? Botulism lives, Clostridium botulinum lives in the soil. They're pretty common.